Hi everyone, my name is Chance Jackson. I am majoring in biomedical engineering in the College of Engineering. My project was to design a novel device for cell imaging, and this was done in collaboration with the Dunlop Group. So thank you to Mary Dunlop and also to my partner for senior design, Tina Ta. So before we jump into specifics and stuff, I want to give a basic overview of what the device accomplishes so that you can kind of get an idea of the pain point that our device is trying to solve. So right here, this is just an image of E. coli cells underneath a wide field microscope. As you can see, you can't really see what's going on. They're very blurry, and that's because they're in 3 dimensions, so the microscope isn't able to focus on all of the dimensions of the cells. On the right-hand side, these cells are now in focus. You can see individualized cells, which is great if you're trying to look at single-cell behavior. And essentially, it's because of our device's main function, which is to inflate down and compress cells. So our device is made of like 2 main parts. There's the bottom, which is just a typical well plate, and then there's the top, which is a pressure chamber. So it's designed in such a way that when the 2 plates mate, if you induce air through a little inlet tube, it's sealed, and then the membrane is flexible, so it will only be able to inflate downwards and into the wells. This causes compression, and then it gives you the ability to get these clear images from otherwise blurry images. All right, so that's where we're going with all of this, but why is this an issue? So in systems and microbiology, you use a lot of bacteria and similar cells. These cells are not adhering, which means they're free-floating in culture. They are not stuck down to the surfaces that they are contained in. In order to image them, however, you need to get them localized to that focal plane. You need them to get to stay in one place, and you also need to get them into 2 dimensions. You can't have them floating on top of each other because that just creates too much noise for your signals, and you won't be able to see what's going on. There are a couple of traditional methods for accomplishing this. The first is using agarose pads. That will cause the cells to dry out over time and in turn affect their physiology and their behavior. That's not good. The other is to use poly-L-lysine. Poly-L-lysine is a substrate that you coat your cover slips with, and studies have shown that it has toxic interactions with cells, which also is not great. Both of these perturb the cell physiologies in some way, which means that the data you're collecting is not necessarily representative of your cells in culture. With experiments, you want to make sure that any changes you're seeing in cell behavior are directly linked to what you're doing to the cells and not the consequence of something like the method that you're using to image them. So that's where we're stepping in with this device. We're trying to eliminate the perturbance of cell physiologies. There have been a couple of ways people have tried to accomplish this same sort of goal. Microfluidics has recently kind of had a big role in this. There are several devices, and depending on what your experiment is, they have different roles. I'm really going to be focusing on this one right here. It's called the Microfluidics Assisted Cell Screening System, or MACS for short. It came out of the Paulson group over at Harvard. And the reason it's of interest to us is because you have 2 flow chambers. You have one for your culture flow that contains your cells, and then you have one on top of it that runs parallel. That is for air. What happens is, when you're ready to image your cells, you induce air into the top chamber, which causes it to expand. The entire device is made of PDMS, polydimethylsiloxane, which is a stretchy elastomer. It's like a rubber. And when the air is pressurized, it kind of acts like a balloon. It swells, and then it compresses the cells underneath down, holding them in place and physically trapping them so that you can image them and see what's going on. There are a couple of issues with this. One, it's not really available in a commercial format. You have to design these PDMS chips yourself, and we wanted to basically take this principle of trapping cells with pressure and put it in a commercial format. So that's where we come in with this project. We are dubbing it the Instapad. I didn't come up with the name. That was already existing when I signed on to this project. But yeah, so essentially, like I mentioned before, it's made of two components. So the bottom is a well plate, and then the top is the pressure chamber. The two will mate in such a way that the membrane can only inflate down into the well, and there's like no lost space for that interaction. It's held together currently by magnetic snap closures, so we just like compress them together. And then there's a ridge along the outside so that we can glue down the PDMS membrane to actually achieve the inflation. And then of course, we have glass on the top and bottom so we can allow light to get through and whatnot. So this is an image of the pressurization process. As you can see, you can't really see what's going on. This is pre-pressurization. You can see that there are cells, but you're not really sure what's going on. Now we're starting to induce pressure by adding air into the top chamber. So everything's going a little crazy. It's a little bit dark, and that's because the microscope needs to focus and it needs to find these cells. So in a couple of seconds, you'll start to see cells come into focus. There they are. Oh, look at that. So now you can see cells, right? If you have a keen eye, you'll notice that the cells that are still floating around are floating past the other cells that are actually trapped. And that means that we have achieved the monolayer. The issue is that some of the cells are smaller than others, so they're able to slide by. Okay, that was the wrong presentation. So, what I did have were some images of our cells with fluorescence. One of the things that we wanted to accomplish was not perturbing the natural cell physiologies. So to do that, we did a propydium iodide stain. That meant that we were staining the cells with a protein that was attaching to DNA, and it wasn't flagging live cells because live cells are intact. There's no degradation of other parts. And that means that everything's where it should be. The protein is not going to fluoresce as much in a live cell as it will in a dead cell. There is an issue of background fluorescence, which we had to account for with our calculations and stuff. But when a cell is live versus when it's dead, there is a distinct difference in fluorescence that we can check. So we ran a propydium iodide stain on our cells under pressurization for the duration of an hour, and we found that about 3% of our cells died over that time period. And we're expecting normal cell death rates, according to literature, to, depending on the E. coli strain and whatnot, be no more than like 5% is what we found in our literature search, which is great. That means that, at least for the time being, our cells aren't being extremely perturbed, and the cell physiology is normal. We do need to do a little bit more testing. We are thinking about looking into other stains. Propydium iodide is only one possible way of checking cell viability and looking at death. We also, in the future, want to make our device more amenable to manufacturing and commercialization. So right now, it is available in a 24 well plate format. It's the exact same size, and it's compatible with all the other stages and stuff that work with the microscope. We're also trying to get it into 48 and 96 well plate format. The problem is that they're just a little bit too expensive, and we want to try and reduce that price point. We also want to add flow chambers so that the device can replenish the culture that's going to the cells, so you can do longer time-lapse experiments, because that would be great if you could look at single-cell behavior over a period of time and see how the same cell is changing. So yeah, that's basically it. Are there any questions? I know I went a little bit over 10, so.
You said expensive. What is expensive?
Yeah, so right now it's 3D printed, and it's being held together by magnets. The magnets are about 10 for like 200 or something. That's not really a problem if you order them in bulk. The real big sting is the 3D printing. So it costs about, I think like $13 to print the top and bottom pieces, which, you know, it's fine while we're prototyping, but it might not be the best when we're trying to, you know, get it into a commercial format. Because then you also have to consider the fact that we need materials to glue the membrane on, and the membrane itself is like, I forget, we got like a square foot of the material, and I think it was like $20 or something at least, which it's definitely not the best. All right, thank you. Sorry again about the slides.